where we're going to hear a talk from Dr. Krista Vandenborne from the University of Florida. Dr. Vandenborne is the director of Imaging NMD, which is a natural history study that's been running for over 15 years, and many of you may have participated in this. And so she's going to talk with us to help us understand a bit more about a measure called fat fraction that we look at through MRI and how this is really being used to help advance our understanding of Duchenne and further drug development. So, Dr. Vandenborne.
All right, well, good afternoon, everybody. It's always such a pleasure to be here at the PPMD conference. One, to see a lot of familiar faces, but I'm always so inspired by the fact that this is a moment where we take almost our collective pulse and see where we're at and bring together families and providers and academic experts and sponsors and advocacy groups to really help move the field forward. So these are my disclosures. The Imaging NMD program is funded by a research grant from the National Institute of Health. So I think it's been pretty amazing over the last couple of days here. We've heard about how much progress has been made in developing the therapeutic pipeline. And now we have several therapeutic approaches that have been approved by the FDA and that are available for patients, particularly in the United States. But we know that we have more work ahead. And so the process, as you know, to move a drug all the way from this early preclinical stage or testing in animal models through the different phases of clinical trials can be a really long process. And there are a number of reasons for them. We know that recruitment and enrollment can be challenging, particularly now when we have so many competing clinical trials. But we also have had a shortage of sensitive measurements that are objective and that can quickly tell whether a drug is effective or not. And so that's been really the focus of Imaging NMD, is to develop imaging-based measures that are sensitive, and that are objective, and that can let us know whether a drug may be effective or not. So the Imaging NMD natural history study, as Eric indicated, was started in 2010 now, so we're at 15 years. Originally, we focused on just Duchenne muscular dystrophy, but we've expanded to also include Becker's muscular dystrophy. We have a total of 403 subjects that have been enrolled. Many of our participants have come from all over the United States, Canada, and even neighboring countries. And as you can see here on the right side, you can see that many of the boys have come to visit as many as 12, 14 times one of our imaging facilities. So as a consequence, we really have this very rich data set, thanks to all of you and all of the families that have made that collective effort. So one of the measurements that have proven to be really valuable with MRI is the quantification of muscle fat fraction. I was getting my cues here up front, and you step back a little bit, so thank you. So muscle fat fraction is a really sensitive measure of disease progression. And just to illustrate this, I've put some images here. These are what's called Dixon fat water images. And so these are images taken of a young boy, 10.5 years old, at baseline of the lower leg, and then we take the same exact image at the same location. And so we see that here on this type of image, fat is shown in green, and muscle or water, which is primarily muscle, is shown in red. And so you can appreciate the increase in fat infiltration just over those 4 years. And so this is just to demonstrate how valuable these measures of muscle fat fraction are as a marker of disease progression. We can quantify the fat infiltration using either an imaging approach, or we can use what we call proton spectroscopy. So when you look at these top images here, you have what's called a fat image in a healthy subject. And so normally the muscle will look black because there's very little fat in a healthy muscle. If you look at the little boy with Duchenne next to it, you could see the white inside of the muscles. And when you look at that from year to year, you can see the progression in that white signal, which is really the replacement of muscle by fat. We can also quantify it using spectroscopy, which is at the bottom. So rather than take an image, we get a spectra. We select that from a specific region. So here you take a voxel, for example, in the soleus, which is one of the calf muscles. And so we have this small signal here in a healthy control. If you look at the lipid, it's really small. And then here the water is, again, mainly muscle. And so typically in a healthy muscle fat fraction is kind of below 5%. If you look at the 10-year-old with Duchenne, you can see this taller peak on the right, some more lipid. We could see the water signal. And then importantly, when you look at it over time, you can see this progressive increase in the lipid signal and a decrease in the water signal as the muscle is replaced by fat. So we have now quantified this in a very large cohort of boys with Duchenne. Like I said, a total of 228 boys with Duchenne. And so we have characterized this longitudinal trajectory very carefully. So here we just look at one-year changes. These are, if you look at the figure here on the left, this is fat fraction measured across different age groups. In yellow, you have healthy controls, again, very low levels, about 5%. And then in blue, you can see with boys with Duchenne, and then 1 year later. So we know we can detect changes in muscle fat fraction, even in young boys as young as 5 or 6 years old, over the course of just 1 year. We have now characterized this trajectory in a large cohort. And so here on the right side, you can see fat fraction again as a function of age, but these are now individual data. So each dot represents a measurement in an individual. And you can see the progressive increase in this fat fraction. You can also appreciate there's a lot of variability, with some boys showing slower progression and other ones showing a faster progression. We've also looked at the ability to maybe measure change in a shorter time frame, and that's important when you're doing clinical trials. The more sensitive we are, we could potentially do shorter trials. So rather than looking at it every 1 year, we've also looked at 3 months and 6 months. And so in this case, each of these bars is a subject or a participant. And you can see that in general, we can detect changes over the course of 6 months, which is a shorter time period than typical functional endpoints. When you're developing measurements, it's not just enough that these measurements are sensitive. Ultimately, they have to be clinically meaningful. So in order to determine that, all of the boys have also done functional tests during each visit. And so here we can see the correlation between performance on the 10-meter walk-run and fat fraction, showing that muscle fat fraction significantly correlates with function. And that is true for all of the functional endpoints that we have taken. Even more importantly, muscle fat fraction is very good at predicting loss of function 12 months or even 24 months later. And so here, just to illustrate that, when there is a 1 standard deviation increase in fat fraction, so if fat fraction increases by just 20%, it increases the odds of losing ambulation over the next 12 months tenfold. So that really means it's an excellent predictor of loss of function. And that's really important when we're trying to develop measurements for clinical trials. At this point, we have also modeled these trajectories, and so that really allows us to quantitatively describe that. So here what you can see, if you look at the red curve, that fat fraction trajectory follows kind of it's a sigmoidal function. So it's slow in the beginning, and then you can see this rapid increase until it slows again, simply because the muscle has been replaced by fat. You can do that for entire, so you can do trajectory modeling for an entire population, for subgroups, maybe a treatment group or a placebo group, or you can do that at an individual level. And so we can look at this inter-individual variability now by capturing something that's called age at half maximum fat fraction. So similar to when we say age of loss of ambulation, that also means something to us. The same thing when we're looking at the age at which half the muscle is replaced by fat. So that allows us to look at people that are maybe more rapidly progressing versus slower progressing, or if there's a shift in this measurement with treatment. So a lot of work has been done in trying to characterize this, to try to understand these measurements. We've also looked at the ability to measure a therapeutic response. And so when we started out the study, it's very different than today, right? We didn't have these drugs that had been approved. So we looked at the effect of corticosteroids. We all knew that corticosteroids slow the rate of disease progression and move the loss of ambulation. So we've looked at the ability of muscle fat fraction measured by MRI to detect the effect of corticosteroids. We've looked at that over short term, over 12 months, as well as over the disease trajectory. So here you have what's called change in fat fraction over 12 months. In orange, you have young boys, the average age was 6 years old, of boys that remained corticosteroid naive over the period of 12 months. And in blue, we have the boys that started corticosteroids. And you can see that when you measure the change or the increase in fat fraction over the course of 12 months, that's much smaller in the boys that were treated with corticosteroids versus the boys that remained corticosteroid naive. We've also looked at the trajectory of fat fraction over the course of the entire study. And here we used this modeling approach again. So we modeled the boys that were enrolled in the study that were corticosteroid naive versus the boys that were on corticosteroids. And what you can see, it's almost a complete rightward shift in this curve. In fact, the age at which half the muscle was replaced by fat in the soleus muscle was shifted by 5 years. So this really demonstrates the ability of MR measures of fat fraction to be responsive to treatment. We've now also looked at the effect of deflazacort compared to prednisone and prednisolone. And you can see, again, there's a shift to the right with deflazacort. And if we look at the age at which half the muscle is replaced by fat, it shifted by 2 years. So based on the observational studies, we've learned a lot. At this point, these MR measures of fat fraction are being used in clinical trials. And so here is a list of some of the clinical trials that have been using these kind of muscle MRI measurements to help with the trial. This has been phase 1, phase 2, and phase 3 trials. And I just wanted to give 2 examples today, I think, of a very effective use of this measurement. So the first study I wanted to describe is the study that was sponsored by Telpharmaca. That was, as we know, Duphasat. As you know, this counteracts downstream effects of missing muscle dystrophin. And so the trial was an 18-month trial. It was a phase 3 double-blinded placebo trial that was done in 11 countries. And so this was a really important use of muscle fat fraction. So in addition to using it as a secondary endpoint, so as a measurement of the treatment efficacy, they also used it as an inclusion criteria. And so essentially, we gave them access to the imaging and MD natural history data to let them determine the proper range for fat fraction as an inclusion criteria. And they wanted to make sure that they selected a cohort for their trial that would not lose ambulation over the course of the 18 months. So they took advantage of this predictive power of the MR measures of muscle fat fraction. And it was very effective because only 1 out of the 120 patients that enrolled lost the ability to ambulate. So MR spectroscopy of fat fraction was used as a secondary endpoint, and the imaging measures of muscle fat fraction were used as exploratory endpoints. We know now that the primary endpoint was met and the drug was approved by the FDA. And so here, what we found that when we looked at muscle fat fraction using both measurements, both methodologies, so both using imaging as well as using spectroscopy, and across the 5 different muscles that we studied, that the treatment group showed a smaller increase in fat fraction than the placebo group, providing objective confirmation that givinostat or Duvyzat ameliorates muscle deterioration in patients with Duchenne muscular dystrophy. The second trial that I think did a really nice job of using these MR measures is the EMBARK trial of Elevidys. As you know, this is a gene transfer therapy in which we produce a truncated or engineered functional form of dystrophin. The trial was done in young boys. These were ages 4 to 8 years old, so little guys. It was also a phase 3 randomized placebo-controlled trial. But in this case, MR was used in a sub-study of 39 participants out of the total group, and it was considered MR was an exploratory endpoint. In addition to measuring fat fraction, they also measure something that's called muscle T2. And muscle T2 can be valuable when you're looking at young boys, because it's not just sensitive to changes in this fat infiltration, it's also sensitive to muscle damage, edema, and inflammation. So we see elevation in this muscle T2 in young boys well before we see functional decline and well before we see changes in fat fraction. So this became an important parameter in this particular trial. So here was the overall study design. So we did MRI measurements at baseline after part 1, the first 52 weeks before the crossover, and then again after part 2. When we compared the treatment and placebo group after this part 1 before we crossed over, we found that the 2 groups at baseline were well matched based on corticosteroid use, also based on all the baseline demographics and clinical characteristics, as well as MR measures. And so when we looked at the results, first for muscle fat fraction, you can see that the magnitude of the change in the treatment group was smaller, again across both methodologies and all of the muscles that we looked at. And when we looked at muscle T2, we found that in the placebo group, similar to what we see in the natural history cohort, there's an increase in muscle T2 in the placebo group, but in the treatment group, we saw a decrease or a stabilization. So since there was only 39 participants, this study was not powered for individual MR parameters, and so a post-hoc global statistical test was done to determine the strength of the treatment effect comparing placebo as well as the treatment group, and they found that there was a significant difference supporting the overall treatment effect benefit of Elevidys on skeletal muscle in this young cohort. So those were 2 examples that I wanted to highlight of how MR measures are being used in current clinical trials. So where are we going next? A big effort right now is to really engage with the regulators. And so we have a large initiative that we've been working on now for the last 4 years. So typically when you're dealing with biomarkers, they can be accepted by regulators as you plan your trial on an individual basis. But our goal as a community has been to really have a larger impact and really help clinical trials across the space. And so in terms of the FDA, they have 2 paths that you can use for biomarkers. One is what's called biomarker qualification, and that's a very long process, and the bar is pretty high. Another process that's relatively newer is what's called fit-for-purpose pathway, and that's really for drug development tools. So that's the path that we chose to move forward. And so this is a collaborative effort with DRISC or CPAT, and we also have a biomarker steering committee that consists of academic experts, advocacy groups, and industry sponsors to really help to bring this initiative forward and take it with a submission to both the FDA and EMA. And so what are we trying to do? Well, what we're trying to do is take advantage of all of these natural history data. So over the last 15 years, it's a very large data set. So we're trying to use the data set. In addition to that, we had clinical trial data that were shared by Eli Lilly, Summit, and Catabases. Bring all these data together, build these very sophisticated models, validate the models, and then build what's called a clinical trial simulation tool. So we can simulate trials before we actually do them to optimize it. And so here's what our current clinical trial simulation tool looks like. So a user can select their criteria. They can select the ages. They can select fat fraction, maybe at baseline, similar to what was done by Pharmaco. They might select certain distances that individuals can walk. They can optimize their inclusion criteria. They can choose their clinical trial design, and then they can run the simulations. And so the goal is really to be able to simulate trials before we invest all of the resources and run the trial. And so that's really the goal, to help optimize these trials that use MR measures of muscle fat fraction. This tool is now publicly available. There's the website for people to use. In terms of our efforts with FDA, we've made a lot of progress over the last 4 years. We have submitted what's called the briefing documents, a very big effort. We have responded to all of their comments. They've reviewed the clinical trial simulation tool. We've shared all of the data with the FDA as well, so they have access to the data. And so we think we're really very close in this initiative. So I just wanted to summarize here that based on all the work that we feel that MR measurements are sensitive, they're objective, and they're responsive to treatment in Duchenne muscular dystrophy. Muscle fat fraction is now being used as secondary and exploratory endpoints, and as you heard earlier too, as an inclusion criteria in multi-center national clinical trials. And then in terms of our engagement with the FDA, we believe our fit-for-purpose application is very close. And our next steps is to include clinical trial data, treatment data, like the data that I shared earlier, to help build a disease drug trial model. And so I just wanted to finish out with this slide here. This was a picture taken at the University of Florida right after approval of Duchenne. And we invited all of the local boys that were part of the Imaging NMD natural history study to celebrate, to really celebrate the pivotal role that they played and their data played in help move drug development for Duchenne muscular dystrophy forward. And I think that can't be overstated. I think it's really important that when people participate in these kind of observational natural history studies, the importance they play in helping with drug development. So I really wanted to make sure that I give a big shout out to everybody that participated and that contributed. And then here are just my final acknowledgements. Again, all of the participants and families that have generously volunteered their time for these studies, and of course, the funding agencies and all of our co-investigators. And I'll gladly take some questions.
Thanks for the talk, Krista. I actually, very encouraged for the work being done on the regulatory front. Maybe a little bit of a different question,
Sure.
as I think a lot of families are used to the use of kind of cardiac MRI as part of routine care. Is there any thought about, the use of this maybe early as part of routine clinical care for decision making in terms of what therapies to use, or even monitoring as we're starting to layer on multiple therapies, given that the sensitivity that we can detect with this?
Yeah. So right now, muscle MRI is not reimbursed. It's not used for clinical care, unfortunately. It's very different than cardiac MRI. So I would love to see that. I think there's a lot of value. I think whenever we talk to families and they see the MRIs, it really, it's very powerful and very informative. But unfortunately, it's not currently part of clinical care. Well, I thank everybody for joining. I know it's the end of the meeting. I think this was the last science talk, so there's a party tonight, so make sure you come.